And we continue sorghum spree with no one else than Charlie Messina from University of Florida. I think he doesn't need any more introduction because everyone knows him. So please.
Thank you very much, Jana. Let me wear for a moment sponsor hat. This is a new institute or center that was funded by growers, in particular at the University and State of Florida, to accelerate genetic gain, particularly in specialty crops with a strong emphasis on human health. So if you're interested in collaborating, you have my phone, email there. A few words about how the center is structured, is core capabilities with a very industrial bias here from concept to a product framework. You can see there's an AI prediction, this leveraging an investment or a donation from NVIDIA, about $80 million on an AI center. We are building these crop transformation facilities with speed breeding and whatnot. We have about 50 breeding programs at the University of Florida. Many of them are fairly small, but strawberry breeding and citrus are of quite a large. We have a field evaluation networks we are expanding for regulated testing that will give us a strong capability to work with gene editing and evaluate in the field what's going on. And finally, we have a nice license in our regulatory that can help us take the products all the way to the growers. Just an example, we are working on citrus, which is a big problem with the greening. We are using AlphaFold to predict receptors, so we can actually trying to fool the plant to how to react to or not react to the presence of the bacteroid. But anyways, that's our first problem since citrus production in Florida went from about 10 million boxes to about 2. So essentially, it's a tremendous impact. Enough for my sponsorship hat. So let's go back to science. And the key question here is trying to understand the rate of genetic gain or lack of it in sorghum in the United States. And thanks for my collaborators, co-authors, and thinkers. A bit of a story started with an observation with a paper we wrote with Laura Mayor. All you need to see there is the green blobs are shrinking. Essentially, if you, at this rate, pretty much sorghum in the US will be an extinct species. So the question was going on in that. So what became a little bit more alarming is that not just the acres was reducing, but the acres being replaced by maize. So essentially, when you start thinking about drought-tolerant sorghum, is being replaced by more drought-tolerant maize. There's a couple of important issues here with the G becoming species altogether, is the loss of this agricultural biodiversity. The second is we are moving into sort of continental scale monoculture. Of course, that is going to reduction in system resilience and, of course, limited adaptation. Of course, with that something in mind, we ran an experiment with Carla and Mark. It was published by Rotundo later on, where we compare side by side over 2 years genotypes commercialized between 1930 for maize and 2020 in maize, and about 1960 to 2020 in sorghum. Hopefully you can see the two columns. We did this under world water conditions, which we expect maize is going to be more productive and the rates higher, which you can see. Look at the 3 drops of water on the top. But under drought, and sort of we have that notion that sorghum was more drought tolerant. And if you look at do you have a laser? Right there, so you see that bump, that's Aquamax. Those are the Aquamax hybrids. And see there's virtually no genetic gain in sorghum, and certainly the yields are much lower. It's almost like 10 tons against about 5. So much for the drought-tolerant sorghum, at least in the United States, which is quite troublesome because essentially this trend of replacing sorghum with corn acres is going to continue. So is that simple? or maybe why is this happening? And to have an approach to this question, essentially we took this framework that we developed with Mark, published in 2020, where you generate this factorial experiment. This kind of cloud has about 1 billion simulations, where we vary different or create different genotypes as a combination of traits. And think about traits of radiation use efficiency, light interception, allocation of carbohydrates to the plant. Think about the framework that Eb presented earlier today, and that is essentially the composition of the genotype is bring diversity or physiological diversity. And we run this in a number of environments in the United States and made different management, planting dates, plant populations, and whatnot. We're trying to generate a wide range of situations of different soils, environmental conditions, patterns, temperatures, and so on. So each dot in that heat map essentially tells you the frequencies of that evapotranspiration by yield combination. So inside each quadrant of that grid, there are many different genotype by environment by management combinations that can lead to that. You can see that the red area is very much where the corn belt resides, at least the dried. And I would just want to notice that there's a significant amount of zeros, about 1 million plus. So essentially maize can actually crash below 400 millimeters quite severely. Not so much for the drought tolerance in maize. So to understand sorghum, what we did was to create a similar cloud with another about billion simulations of sorghum, varying similar traits, where is the light interception, photoperiod sensitivity, trying to adapt that flowering time and maturity to the western corn belt. So this is a cloud that just sits on the areas west of Kansas and very much Kansas and Oklahoma and Texas. The first thing you can notice is the differences in scale. Essentially, we said corn was much higher. Think about 20 tons yield potentials to 14. The other bit you can see is for those between 200 and 400, there are virtually zero crashes on sorghum, and that's where I think the drought tolerance start coming in, less about total productivity, but the frequency of sorghum crashing relative to maize. So essentially, it's not as much as total productivity, but it's about risk. And of course, maybe part of the shift in the land allocated to corn, it may be a distortion from policies or maybe something else altogether. So anyways, now we have our cloud, and let's see how this sort of methodology can help us understand what's going on with sorghum. So we went and find a number of studies. The first one that we look at came from Rotundo, which is the experiment we conducted in Veluco, and they start suggesting that maybe sorghum has reached a biophysical limit. So if you look at that, the dots that underpin, and I show you the lack of rates of genetic gain, you can see that many of the modern genotypes, they're just hitting that biophysical boundary. But that's the environmental managed environment. So we went and overlay also the studies from Laura and DeMarco. So these were conducted in the Midwest, in that sort of Kansas region, and they sit a little bit, you have a little bit of a gap, but it's between the quantile 80% and 99% quantile. So essentially we are sort of indicating another
of our vegetable breeding activities.
There you go. We even go to different regions. These are public breeding programs, one from Texas, the other one from Nebraska. And you can see they are lower on the evaluation. They're even inbreds in here. They're still sitting above the 80% quantile. So essentially what I think is happening is that indeed these breeding programs are sort of hitting that biophysical limit given the germplasm that is available for the breeders to work with. And this is not just one study, but regardless of which germplasm are using, they are having a very common foundation. There's nowhere to go to make more genetic gain. So if you think about where these programs are, essentially they are sort of circling or revolving around this local adaptation peak. So if you think about a hybrid commercialized in 2003, you can see that slope there, or 2020, you can see it's more sensitive. So essentially they are just trading. So one year they select for more drought tolerance, as maybe one of the head breeders push pressure to make more progress. Sure, they can lift and make progress, but they lose the drought tolerance. Essentially, they cannot lift the both ends as happening in the Aquamax. Essentially, this severe limitation, I think, is a bottleneck on where these breeding programs can go. I'm trying to make it on time. So what can we do to get out of this sort of hole that the maize program, or the US maize program is? And to that end, essentially what we did is look at different traits, whether it's grain field duration, flowering time, carbon allocation, roots. That's, I think there's an opportunity there, or radiation use efficiency. And one of the things that we found out is when you look at, when you conduct a mixed model analysis and estimate the variance components and how relevant are each of these traits to move that germplasm up, most, and remember for 300 to 400, sort of this area here is the sorghum TPE. What you end up doing is that most of the variation is explained by the interactions between traits. And each trait, depending on the environment, of course, radiation efficiency will play a significant role under well water conditions, but much lower when going to drought stress. So essentially, there's no clear patterns here as what is the direction. So essentially, it's a combination of all these traits that makes things even more complicated to drive genetic gain in sorghum. So to conclude, I think it's imperative to do more, but not just more, as one of the big breeders says, well, genetic gain in maize is higher because we put more money in it. It's not just putting more money in this particular germplasm. We need to change that germplasm and bring diversity for adaptive traits. And it's the complexity of this system, as I showed, the interactions underpinning adaptation, that makes breeding for sorghum much more difficult than what you can breed for maize. If you think about what we learned from maize, essentially it was apical dominance and making sure that you can pack more plants per square meter. Apical dominance in sorghum, already the panicle is on the top, so there's not much where to go in there. I think roots could be another problem, another opportunity, as we show in maize, the radiation efficiency, maybe just more allocation of the mass that is sink in the ground, just put it on the aerial parts and in the ear. However, there's a long way to go. And finally, what are the implications of this? Hopefully this differential rate between sorghum and maize will continue shifting land use unless we do something about it. The loss of agricultural biodiversity is troubling for me. Breeding for climate change, which essentially is going to require shifting or combinations of crops, will require systems level thinking. I'm really pleased to see today someone advocating for systems breeding. And therefore, it's going to require investment, not just in maize and soybeans, but in many other crops, so you can start putting all the components together. Thank you very much.
You are perfectly on time. Thank you, Charlie.
See,
so that means also we are perfectly fine with so many questions. So please start.
So it's very interesting that sorghum doesn't fail where maize fails at low water availability. Yep. Do you have any idea about the ecophysiological mechanism that makes the difference between these two species?
I think it's just water conservation. The sorghum grows less, so it saves water, kind of the Aquamax type of. If you look at radiation efficiency, corn has been growing up to 2, sorghum is at 1.3, 4. Yeah. So it doesn't use that much water. Yeah. The other piece is, I can't remember, Amir says there are more roots. The root systems are way bigger. We did trenches in, well, Carla dig the trenches. And sorghum roots can go into the alley and conquer the alley. Well, maize, they're more skinny. So I think there's part of the underpinnings of crop improvement in maize, it came from mass that was in the roots and put in their part. And sorghum still down there.
Yeah, interesting.
So that the ability of capture water has a penalty.
What do you think the relative contribution of heterosis is between the two breeding programs in drought?
No idea. Mark?
You see, and you showed some of the inbred lines in sorghum, and they look like the hybrids. And so obviously, yeah, it just seems like that might be in Aquamax, I assume. Heterosis is playing a massive role.
No more so.
Maybe it's here.
The heterotic groups we were working with when we developed the suite of products under Aquamax, same heterotic groups that exist for all of the temperate maize in the breeding program. So as a breeder, we don't select for heterosis. I mean, it's really the investment goes into building your heterotic groups, and then you just get on with breeding. I think there's been a lot more effort in setting up the foundational heterotic groups in maize, and there's a bit more restriction and sort of the diversity and the amount of work that's gone into setting up the diversity that's in the heterotic groups in sorghum. So I think that's part of how that diversity is distributed and how the breeding programs can access and use it.
But yeah, related to the inbreds and hybrids, that's from the Texas public breeding program, which probably the heterosis is not near as large as the sorghum commercial. And again, there's a lot of points overlap together, but you can see the delta. Commercial breeding is way closer to the upper bound than the public ones. But still, they're in that sort of 80 to 19%.
Hi. Related to that question, I was wondering if there are any wild relatives to sorghum or land races that could be used to increase the germplasm. Do we know of this?
I think we need to look at the work that David Jordan did, and in the Australian germplasm, they have a nice physiological characterization, far better than I think whatever have in the US. And looking at the traits that are segregating there and how we can leverage those to improve or at least expand the diversity, functional diversity in that program. So I think I will start in David Jordan's work.
So thanks so much for your talk. I think maybe following up on what Hendrik had mentioned too, I was curious if there was any sort of characterization of stomatal density and patterning with these lines, and if maybe that's a way to overcome this dip in RUE at these lower, under these drought conditions.
Do you say spacing? Or
Yeah. So density and also patterning.
Sorghum is just tillers.
Yeah, but I think
in maize you have more chances to play with that, even though more modern germplasm, if you let it, is going to tiller.
No, sorry, for stomata though.
Oh, for stomata.
Stomata, yeah.
I think the control of transpiration is at this canopy level rather than stomata. We have that conversation in crops and trying to tweak the conductance. Mm-hmm. Limited transpiration has a role. I think Jana showed it. But the control of transpiration comes from how many tillers, how many plants you have in that field.
Okay, thank you.
The leaf area is going to, that's a big knob on water use, right?
Any more questions? So we are probably done then. You have been amazing. Please give a hand to all our speakers once again. Let's have coffee.
Thank you.